Hello, welcome to Biology Made Easy. Today we are going to look at plant tissues, flowering plant tissues. We are going to discuss how the tissues are formed and how the various tissues can be recognized in sections and their functions. Welcome. Learning objectives. We want to quickly revise monocots and dicot external structural and similarities and differences, list the basic plant tissues, 4 of them, explain the origin of the meristematic tissue, how meristematic tissues forms the other tissues, and identify dicot and monocot stems and roots in plant section. We should be able to list the 3 substances that impregnate or are laid on primary cell walls. We should be able to explain how cell walls are thickened with cellulose or lignin and explain how pits are formed, and so therefore identify the different plant tissues and their functions. Welcome. Flowering plants, as the name goes, have very beautiful flowers. That is one of their identifications. And then flowering plants belong to the phylum Angiospermophyta, class Dicot and Monocots. And then we want to look at their similarities. You see that Dicots and Monocots have basic important similarities in common. Roots, they have roots. Each of Dicots have stems, Monocots have stems. They have leaves, they have flowers, dicots have flowers, monocots have flowers. Basic similarities. Differences, we can use this drawing to work out the differences. The differences are only in roots, taproots, fibrous roots, leaves. You have usually broad, usually narrow. Let's look at their venation. You have reticulate venation in dicots, parallel venation in monocots. You can have longitudinal, and you can also have transverse parallels. Then in leaf differences, dicots have petioles, monocots have leaf sheath that winds around the stem. That's a leaf sheath here. It has leaf ligulus that are absent in dicots. Well, what about flowers? There's a dicot flower, monocot flower, which is usually an inflorescence. Monocot flowers. You see the dicot flowers are usually brightly colored. The monocots are usually dull. They have a lot of bracts, and these are usually insects or animals or water pollinated, but monocots are usually wind pollinators. Look at their flowering parts. Flower parts, dicots usually have 5 or multiples of 5. So you have petals, 1, 2, 3, 4, 5. Monocots usually have flower parts of 3 in 3s. Okay, there are other differences, details that we would look at. Now, in description, both dicots and monocots, dicots, we have shoot system, root system, or we can look at it as the vegetative part and reproductive part. In monocots also, this is the root system and the shoot system. Or we can look at it as the vegetative part here and the reproductive part, flowering. So monocots and dicots basically are very, very similar. Their differences is on how the different basic structures are arranged. In the same way, in plant tissues, in both monocots and dicots, there are 4 basic plant tissues. We have meristematic tissues, or let's look at the cells first. Meristematic cell, you have parenchyma cell, collenchyma, sclerenchyma. And sclerenchyma, we have fibres or sclereids or stone cells. Well, in parenchyma cells, you have modified or parenchyma-like. You have the epidermis, endodermis, pericycle, sieve tube elements, chlorenchyma, Aerenchyma, Koch cells, medullary rays. All these are modified parenchyma. Then you have modified sclerenchyma. You can look at tracheates and Xylem Vessel element? Well, we want to also tell ourselves that lignin, which is the substance that is laid on Sclerenchyma cells, lignin stains red with fluoroglucinol stain or yellow with aniline chloride. So that when you see slides that are stained red, the red parts are Sclerenchyma. Let us look at meristematic cell. All these other cells, Parenchyma, Chlorenchyma, Sclerenchyma, and their modified types, they all originated from meristematic cells. So let's look at meristematic. The meristematic tissue, right? These are meristematic cells, which are formed meristematic tissue. The cells are cuboid. The cells have very thin cellulose cell walls. And anywhere that you have cellulose cell walls, you also have hemicellulose and pectin. Now, these meristematics have a very thin cellulose cell walls. They are filled with cytoplasm and large nuclei, right? Each of them has one nucleus, large, and very tiny or even non-existent vacuoles. And the cells are closely packed together. If you look at this is a shoot tip. Look at the cells. These are all meristematic cells. This is a nucleus. They are tightly packed together. No intercellular spaces. This is a root tip. Also, the cells are quite closely packed together with nuclei. This is one nucleus, that is the cell. That is meristematic tissue. No intercellular spaces. So if you draw a meristematic tissue, this is right. This is not it, because there's an intercellular air space here, which is not found in meristematic tissue. So if you see a tissue like this with an intercellular air space, it is not a meristematic tissue. These tissue, this is an air space here. So you know they are not meristematic parenchyma. They are. We'll look at them. Now we want to look at the origin of meristematic cells that produces all the other cells. Now we have this, the origin of meristematic cells comes from the embryo of the seed. So we have, this is a seed, cowpea seed. We have opened it up. This is the embryo. Here is the radicle. Here is the plumule. These are meristematic tissue. And these plumule is the embryonic shoot. It's the meristematic tissue that will form the shoots. The radicle is the embryonic roots. The meristematic cells that will form the roots. So I drew this neatly here. Plumule becomes the shoot. Radicle becomes the root system. Now, how does the different cells originate from the meristematic cells? We want to look at this. The meristematic cells in the radicle or plumule, or at the root tip or at the shoot tip, that is the apical meristems, are called initials. Well, why are they called initials? All the other cells, tissues, come from them. This is a meristematic cell. We want to demonstrate how they do it. This meristematic cell, it divides mitotically. It means it divides into two identical cells. One of the cell remains meristematic and the other cell will differentiate. Now, the one that remains meristematic, they remain meristematic at different parts of the plant, at the shoot tip, at the root tip, axillary bud, intercalary meristems, pericycle of the root, vascular cambium, and later cork cambium. Now, the cell that differentiates starts by elongating. So it elongates and expands by absorbing water by osmosis to form more vacuoles and large vacuoles. And the vacuoles or coalesce, they fuse together to form bigger vacuoles, while the cellulose cell wall are taking in more cellulose, expanding, stretching, and then the walls become impregnated. Some of the walls get thickened with cellulose, and some of the walls get thickened with lignin to form the parenchyma, collenchyma, and sclerenchyma. Now, we want to look at the position of the meristematic tissues in the plant. We have meristematic tissue at the shoot tip and at the root tip. These are the apical meristems, and these are the initial. Then some meristematic tissue is left in the axillary bud, intercalary meristem at the node, some in the vascular cambium, some in cambium, some at pericycle at the root tip. So now we want to look at how the various tissues are formed from the apical meristems, right? Now, this is the root tip with meristematic tissue. This is another root tip. Now, this is the shoot tip. We have apical meristem here. All these are going to form leaves, and these are leaves, very tiny leaves that form around the apical meristem to form a bud. Well, here is also another shoot tip. We have this going to form a leaf primordium. Then the apical meristem is here, but all here are also meristematic tissue produced from the apical meristem. Now, let's look at how these apical meristem produce all the other tissues, the parenchyma, collenchyma, sclerenchyma. So how the various tissues are formed from the apical meristem. We have this as a plant. This part is the shoot or the stem, and this part is the root. Just an illustration. Now, the apical meristem at the shoot tip is here. The one at the root tip is here. And at the root tip, this place is called the quiescent zone. These are meristematic cells that are dividing very slowly, and they form the apical meristem here, and then the root cap, which is parenchyma. Now, the apical meristem here divides and forms this structure called the protoderm, and forms another place called ground meristem, and procambium here, and another ground meristem. Now, these are all meristematic. So this zone we can call the meristematic zone. All right. Then we have a zone as the cells grow, they elongate, they expand. So we have a region of cell elongation and expansion, and then they continue growing and expansion, and then differentiate, take on new shapes, sizes, structures, lignin or more cellulose, and form different tissues. So they differentiate to form different cells. So you have this protoderm form the epidermis. Ground meristem forms the cortex. Another ground meristem inside forms the pith. And then the procambium form the vascular tissue. This is happening in the roots. In the stem, the same thing is happening at the shoot tip, producing protoderm here, ground meristem, procambium. The protoderm elongates, differentiates to become epidermis. The ground meristem becomes the cortex, and then the pith, and then the vascular procambium forms the vascular tissue. So this is how the apical meristems, the initial, produce all the other tissue. Well, epidermis is parenchyma, cortex is parenchyma and some collenchyma, and then the vascular tissue is sclerenchyma with some parenchyma. Well, let's look at how these look in longitudinal section. But before then, in the roots, you can also have the procambium filling the whole middle here, and then the vascular bundle is filling the whole space here. That is in dicot roots. We'll look at it further. But let's look at these tissues: epidermis, cortex, vascular tissue, and pith. Well, a longitudinal section is illustration of a monocot root. For instance, you have the epidermis, and the epidermis is parenchyma. You have cortex, the cortex is parenchyma. It can also be collenchyma, we'll explain. Then you have a portion called endodermis, which, as we have said, is a modified parenchyma. You have the pericycle, it's a modified parenchyma or meristematic. Then you have the vascular tissue, which is sclerenchyma and some parenchyma. Then you have pith, which is parenchyma. So it's still the 4 basic type of cells. Now we want to look at the tissues, as you can see in a primary tissue, that is plant parts that are not lignified or gone through secondary thickening. They are soft, they are delicate, they are still growing. Here is the primary tissues and transverse section. This has been produced so that when you see a dicot stem section, you should see it. That is the dicot stem. Look at how the vascular tissues, bundles are arranged. All right? All right? When you see a monocot stem, you should see the vascular bundles are scattered. If you see a dicot stem, the vascular bundles are in the middle, xylem filling the middle. If you see a monocot stem, the vascular bundles are arranged in a kind of a ring here. Well, if you look at the dicot stem, the epidermis, we have talked about it, is parenchyma. You have the cortex. When the plant is young, the cortex here, you can have collenchyma and parenchyma. Then you have this zone that we term pericycle, is sclerenchyma. And then here, in between these vascular bundles, you can have parenchyma called medullary ray. We'll look at them. Good. So this has been produced so that you can see the various tissues easily. When you see them, you should know I'm dealing with dicots, monocots, stems, dicots, roots, young roots, monocots, roots, young roots. Good. So we want to look at the details of the various tissue. But before then, this is a dicot stem, a young one. You see here is Sclerenchyma because it stains red. All right, good. And this Sclerenchyma is all around the plant, the stem, when it's young. You can also have the Sclerenchyma just as cups. All right, it forms just a cup. All right, below the vascular region. This is the vascular region. All right, okay. Xylem is here, phloem is here, cambium is here. You can have it like this Sclerenchyma here. Good. You can also have the Sclerenchyma all around the vascular bundle. Like here, you have Sclerenchyma all around it. Good. Now, we'll look at these tissues again. But before we look at the detail of each of these tissues, we want to see how cells, the primary cell wall, can be impregnated either with cellulose or with lignin. So we want to look at the 3 substances that impregnate cell walls and change the cell walls. They are lignin, suberin, and cutin. Now, lignin is a complex polymer, hard, all right? And it gives tensile strength, meaning the plant tissue does not break when it's stretching. Compressional strength, the plant tissue does not squeeze or crumple under pressure. So lignin forms a barrier around plant cells, all right? And lignin is formed in Sclerenchyma tissues, right? And the cell dies. So it forms a barrier that protects the tissue from pests and others. Then you have suberin, which is also another substance that impregnates cell walls. It's waxy, all right, layer that is laid on the primary cell wall. It makes the tissues impermeable to water and gases. Also, it seals tissue after wounding or leaf fall. Then you have, that's a leaf fall, leaf abscission. Then you have cutin, which is also like cutin, a waxy substance, and it forms over epidermis of shoots and the root cap to protect them from desiccation, losing too much water, and also from pests or injuries, all right, especially the root cap as it penetrates the soil, and it also constrains stretch of cells. Now, we want to look at tissues, the detail of each of these tissues. And first of all, we want to look at them as supporting tissues. So what are the supporting plant tissues? We have turgid parenchyma, collenchyma, sclerenchyma fibres, Scleries, Xylem Vessel, and trachees. These are the plant tissues that give strength and support to plants. Now, let's look at turgid parenchyma. Now, this is a generalized parenchyma cell drawn. We have one cell here, one cell here. We want to quickly tell ourselves of the cell membrane, cell wall of this cell here, cell membrane of this cell here, cell wall of this cell here. You have the middle lamella separating them. You have this plasmodesmata, plasmodesma, all of them plural. You have plasmodesmata. And filled with cytoplasm, you have the nucleus here, nucleus, and a large vacuole. This is a characteristic parenchyma cells. And these plasmodesma is a gap that has cytoplasmic strands. So cytoplasm streams from one cell to another through these plasmodesmata. And parenchyma cells are able to absorb a lot of water by osmosis, and the cell becomes filled with water and turgid, meaning tough and solid, all right? And by that, it's able to maintain strength, give strength to herbaceous parts of the plant. Herbaceous parts is the soft parts that are not thickened. We'll look at this again, but let us look at collenchyma tissues. Collenchyma tissues, well, they are also living. See, the cells have cytoplasm, they have nucleus, they have vacuoles, just like parenchyma cells, but collenchyma cells have extra thickening in their radial walls. So this LS, this wall is thickened. This wall is thin. So extra cellulose on their radial walls. The TS, you have this area is thickened and this area is thin. Now, collenchyma tissue have very few intercellular spaces, all right? We want to look at how you can easily see collenchyma tissues in plant sections. Well, when unstained, the wall looks glistening, and methylene blue stains the tissues dark, make them stand out dark. Now, let's look at it. If you have a plant section, how do you work out that this section is collenchyma? You see, here is the plant section. Here is the epidermis. Here is collenchyma. Look at this. This wall is thick. This wall is thin. You see, this is thick, this is thin. They have irregular thickened with cellulose. And as we said, anywhere we have cellulose, we also have hemicellulose and pectin. So you can easily see that this is collenchyma. It's different from this area, which is parenchyma. Thin walls, intercellular spaces. So you can also work out the difference between collenchyma and sclerenchyma in plant sections. So take note of this. Collenchyma. Good. Now, where are collenchyma tissues found? As we have said, together with turgid parenchyma, they give strength and support to tissues. So, collenchyma is found in growing plants very close to the epidermis, right beneath the epidermis. So the epidermis is here, and collenchyma is here. And here is parenchyma. Here is sclerenchyma. Of course, they stain red. All right, good. So that is the very close under epidermis in growing plants, that's herbaceous plants. Then also, they are found as strengthening tissues in veins, leaves. And so they are found as a close knit of cells around the vascular tissue of leaves. The vein, the vascular tissues are in the veins, are the veins. So if you look at this section we have here is collenchyma. It's called bundle sheath because it's just one layer of neatly closed cells, all right? And it can stain blue-black with iodine because it contains starch grains. So it's called bundle sheath around the vascular. This is xylem, this is phloem, and here is the collenchyma. And this whole thing is the vein. Here is the vein in the leaf, and collenchyma around the vein to support the vein. Now, we want to look at xylem now. Xylem fibers, sclereids, xylem vessels, and trachees. But before we look at the cells, we want to find out how the cells are thickened. The primary cell wall is thickened with lignin, and pits are formed because you have pits in all these. All right. So we have these two cells here. This is we want to look at how the primary cell wall is lignified, all right, with a secondary wall. So we have this cell again to show the plasmodesma, another one here, and the cell. Now, lignin is laid on plant cells starting from outside the cell wall, very close to the cell membrane, all right? So the lignin is laid, and more lignin can be laid, and more lignin laid, and the cell dies, and a lumen is formed. All right, so we have lignin laid, but the lignin is not laid where the plasmodesma are. This is a plasmodesma. The lignin is laid all over, but not where the plasmodesma are. So where the plasmodesma are, where lignin is not laid on the primary cell wall, the plasmodesmata, a group of them very close to each other, form what is known as the pits. So close group of plasmodesmata forms a pit field, all right? So this one is the plasmodesmata. And look at the primary wall, one primary wall, and this is the middle lamella in the middle there. And lignin is going to be laid here, and the rest of the cytoplasm is up there. So this is how it is. You have the simple pit cavity. This is the pit, simple pit. Simple because the distance in there is the same. The tubes are the same width. The same width here, the same width here, the same width there. That is why it's called simple pit. The tubes are the same width, right? This tube and this tube, the same width. Now, this lignin is the secondary wall of lignin that was laid, all right? And this is how simple pits are formed. Now, we want to say that we also have another type of pit. This is a bordered pit. This one, the lignin is laid in such a way that there are flanks of the lignin. This has a flank here. This has a flank here. Flanks, all right? And these flanks, borders, there's a thickening here called torus. And this thickening can close. It serves as a valve that can close the whole pit. Okay? Now, in the simple pits, the lignin is laid before the cell wall got thickened with cellulose. So the area where the lignin is, the primary cell walls are thin. Okay, well, this is also a bordered pit in electron micrograph, shown with electron micrograph. Good. Now, we want to say that in the laying of lignin to form pits and to form sclerenchyma tissue, in fibers, the lignin is laid all around, thick around the cell, except where the plasmodesmata are. All right? It's laid all around. So you have a solid cell wall, secondary cell wall of lignin, and this becomes a lumen, dead cell. In other cells, like the xylem vessels, the lignin is not laid all over the cell. The lignin is laid in various tiles all over the cells. All right? So in plants, we have fibers that the lignin is laid all over. And here is a lumen. It's a lumen. Here is a pit. And this cell is completely dead. Well, when it's dead, the pits have no function at all. When you look at the xylem vessels, the lignin, the secondary wall formation, this is lignin, laid in some annular kind of rings. So here's the primary wall, still there. Here, the lignin is laid in a kind of a spiral, so it's called spiral thickening. Here, the lignin is laid in a kind, a spiral when it stretches, all right? So the lignin laying is different in different sclerenchyma tissues. Well, it's enough for the time being, so we go on. Let's look at sclerenchyma fibers and how you can recognize them in sections. Now, as you can see, the fiber is long, pointed cell. They are dead. They have only lumen, tapering ends. Look at in LS here. You have, these are various cells. This is one cell. You have the pit here, various pits. You have lumen, empty space because of the lignified wall. This is how you see it in LS, sclerenchyma tissue. In TS, you see very tough walls, all right? And you can see pits joining one lumen, it's one lumen, and you can see the pits joining them and the lignified secondary wall, very thick. Now, let's look at how they are arranged. They are arranged end to end, all right? The end walls interlock so that you have long fibers, strong, and it increases their strength. They interlock to increase their joint strength. They can also be laid as sheath, and also that gives mechanical strength. So wherever these sclerenchyma tissues are, mechanical strength is what they are doing to the tissue. Now, we want to look at these fibers in section so that when you see them in section, plant section, you should be able to recognize them. You saw how the collenchyma was, all right? The thickening here, look at all this is thickening, secondary wall. This is lumen, this is lumen, they are all lumen. But then, besides the thickening that is characteristic of sclerenchyma, we look at it very closely, you can see pits. There's a pit here between this lumen and lumen. Look at this pit. Very neat pit here. There's another pit here. All right. That is sclerenchyma. And where they are also, their position also gives them up. Very thickened wall. All these walls are thickened with lumen. Okay, good. Now, you look at this one, this black and white. So you look at this one, you can easily see this thickening, very thick. And they are around this vascular bundle. See, very thick and red. If you look at it very carefully, we should see a pit somewhere in some of the sections. Here, we don't see any pit easily, but let's come here. This is also Sclerenchyma. This one is part of a pit here. Here, part of a pit here. And this is all Sclerenchyma, very thick walls, right? So this is how you notice Sclerenchyma in plant sections, all right? Now, we want to look at where you can find them. As we have said, they give strength to growing plants, so they are close to the epidermis, the cortex. They also are found around vascular bundles to give strength to the xylem and the phloem cylinder. Xylem is conducting water, phloem is conducting food. They need strength, so Sclerenchyma tissue strengthen the area. Here's another Sclerenchyma tissue strengthening the area as a cup. All right, good. So that is where they are found. Good. Here is also Sclerenchyma around the vascular bundles of monocots all around it to give it strength, not so that the tissues will not collapse. Now, Sclerenchyma, we also have Scleries. Scleries or stone cells are also roughly elongated cells. They are also lignified, but the lignin in Sclereids is laid in concentric rings. Concentric rings, it starts from outside the cell. This is one sclereid here, another sclereid. Here is the lumen. Here is the lumen. This lignin started moving, concentric rings, and the lumen are left. All these are pits, all right? All right, so you see this, you have some pits, and then the lumen. These scleries or stone cells are found in the cortex or pits, are found in the cortex or phloem of fruits and seeds, and mostly, all right, in fruits and seeds, but they can also be found in some stems, and they give the gritty feeling to fruits and seeds or any part of the plant where they are. That's Sclerenchyma or stone cells. Well, now we want to look at xylem vessels. And xylem vessels, we want to first of all look at how a vessel is formed. The xylem vessel, long tube, is formed from cells. These cells are elongated, tapering ends, all right? And one cell is called the xylem vessel element. So you have one xylem vessel element, another one joined to form a long tube. That's a xylem vessel. And the vessel, this xylem, this is element, this element, and this is element. These are their cross walls. Cross wall. And these cross walls break down. And when they break down, then you have a long tube with a long hole to carry water. Now, these cross walls here, they break down in the center so that there's a hole in the center. This to illustrate it, all right? This how it really is. This how it is illustrated. The hole, you have a rim here and a rim here and the hole. Well, the walls are lignified. The cross walls are also lignified. So there's something like this, all right, happening. Then this hole for water to conduct through it. Now, this is a xylem vessel. This is the hole in which the water goes through. Here is the wall. And the walls, as we have said, the lignin is laid down in various factions: concentric rings, annular thickening, spiral thickening at the beginning, and later spiral thickening is like this. Now, these cells, these xylem vessel elements, they are called protoxylem. The thickening is formed when the cells are growing at growing parts of the plant. All right. So the cells are still stretching, and they are called protoxylem, and they have this kind of characteristic thickening. Okay. These are produced in cells still stretching. And these ones, metaxylem, this is a kind of thickening, secondary thickening. You have scalariform, they are laid as like steps, all right? And then you have reticulate thickening. The reticulate is these steps being joined to form a network of thickening. Meanwhile, the primary cell wall is still around, okay? Then you have this lignified secondary wall, leaving only pits. So this one is pitted, very strongly lignified. These are found in old parts of the plant, grown parts of the plant, and these cannot stretch anymore, cells that cannot stretch, and they are all dead with lumen, all right? And the cells are greatly lignified, leaving small, very small or no areas of primary cell wall. You have a primary cell wall here, primary cell here, no primary cell, all pitted, okay? And by the time these ones are formed, the plant has stopped, the area of that plant has stopped stretching, all right? And these cells are all dead. They have lumen. These ones that are still stretching, the cells are not really dead. And so there will be small cellular conduction flow between the pits here. But these ones, no, the cells are dead and the pits are valueless. Now we have the xylem tissues, a few here. We can see this one is like annular thickening, isn't it? This. Well, we'll see more of them better in better slides, but you can see them, all right, that these are xylem vessels. This one is, a lumen is showing, this is part of the cell wall that we are seeing, but this part is the lumen. Good. Now, here are still xylem vessels. You can see this one is like spiral. Here is some. They look spiral or scalariform. Look at this one. This is the lumen, great lumen, large lumen. All right. These are pits. These are all pits. So these are all thick secondary walls. All right. So this is how xylem vessels can be seen in LS. Okay. Longitudinal section. Right. Now, we want to look at xylem vessels as you can see them in TS. Well, you see, this is Sclerenchyma. All right. Here is xylem. The xylem vessels have very large lumen. All right. Large lumen. Larger lumen than the Sclerenchyma. So you can't miss xylem vessels. Here in this one, all these are xylem vessels. Vessels. Around are other tissues that together form the xylem. Trachees, we'll talk about them. All right, good. We'll come back to this. Remember, we said this is meristematic cambium. Right. Now, let's look at the trachees. The trachees, the cell looks elongated, tapering ends, not as tapering as the xylem, not as tapering as fibers, but they are also joined end to end like xylem vessels or fibers. All right. The cells of tracheates are also lignified, and they also form scleriform, reticulate, or pitted thickening like xylem vessels. When not mature, they are not dead. Just like xylem vessels, when they are not matured, they are not dead. The cytoplasmic streaming and through the pits or through the plasmodesmata. All right, but when they are dead, they have a lumen. And tracheates are very common in conifers. They are the conducting tissues, the main conducting tubes or tissues in conifers. They are not too common in angiosperms, but they are also part of Sclerenchyma in angiosperms. Now, we want to now look at parenchyma tissue. We've been talking about parenchyma tissues quite a lot. Well, the cells are not specialized. They are living. They have cellulose cell walls. And cellulose, it means they also have hemicellulose and pectin. They have large vacuoles with cell sap. And commonly, intercellular spaces present. So when you look at this one, of course, these are, they can be nothing else but parenchyma. Look at their thin walls. Look at the intercellular spaces. This is parenchyma. Look here, we have collenchyma. Look at irregular thickening of the cells. Thick, thin, thick, thin, thick. So here is collenchyma. All right. Okay. We hardly see any intercellular spaces. All right. There are very few intercellular spaces. Probably this one looks like an intercellular space here. All right. But let's look at this. Here is parenchyma. See the thin walls compared to the collenchyma walls. This is sclerenchyma, thin walls. Look at the intercellular spaces. One here, one here, one here. So this is parenchyma. Cells filled with contained cytoplasm, large vacuoles. Tissue has intercellular spaces. You can't miss them. Okay. Now, we want to look at functions of the parenchyma tissues. Plenty. We talked about parenchyma as turgid parenchyma as a strengthening tissue with collenchyma in primary tissues of plants. When the plant is still growing, it hasn't thickened yet, it's herbaceous. Then you have turgid parenchyma and collenchyma as strengthening tissue. Then they can form packing tissue. We talk about them. They also do lateral conduction through medullary rays. They have endodermis. They have intercellular spaces, so they do gaseous exchange. You have some Aerenchyma, all right? Mainly parenchyma cells with large air spaces. Then they also serve as epidermis. They have structures that store waste. The parenchyma cells store food and water. They also serve as pericycle sometimes, Koch cells, and then photosynthetic tissue, all right, with chlorophyll to photosynthesize. So, well, let's look at the packing tissues. You see, packing tissue means that anywhere that there's space to fill, parenchyma cells will fill the place. This place is all parenchyma. See, there's a hole here. It can be filled with parenchyma, all right? So you have this xylem tissue here. It's mainly xylem. We saw them. These are all lignified walls. You have these thickenings, characteristic of xylem. You have this hole. Look at this wall. These are xylem. You see, this one says xylem, xylem, xylem. There's no thickening that you can see. And it's around, it's in where the xylem is. So it's a packing tissue, filling space. Now, when you come to cells that are neatly arranged, you can have xylem filling a space like this. Well, so these are called medullary rays. This one is a xylem. All this is secondary thickening, thick wood. But this place, this place is parenchyma, packing tissue, parenchyma. Medullary rays, these are them, parenchyma, packing tissue. Amidst these thickened wood, secondary xylem. All right. So that is why they are called packing tissue. All right. Now, they also do lateral conduction of water through medullary rays. You see, this area is wood, secondary xylem, dead. Much of the cells are dead because all these have lumen. They are dead with sclerenchyma all around it to toughen the tissue. But this is a layer or layers of parenchyma. All right, and those are the medullary rays here. So there's lateral conduction of water. Water moves from one cell to one cell across here, across the medullary rays, so that the cells here are always alive, keeping the plant tissue alive. So lateral conduction through medullary rays. Here are medullary rays, and there will be lateral conduction of water. Lateral conduction. Water moves across like this. Okay, and through that's parenchyma, very neat. Now you also have parenchyma tissue serving as the endodermis. Well, we have packed a lot here. Let's look at this first. This is endodermis. The endodermis tissue can be just one layer of cells thick, close-fitting cells. Close-fitting cells. It can also be two or more layers. But in roots, it's one layer of cells thick in roots. Okay? So these endodermal cells, they have their radial walls thickened with suberin. So this is the cell, the tissue. This cell and this cell, you have suberin, 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 suberin. Radial walls thickened with suberin. Suberin does not allow water or gases to pass through. Okay, so this is a young root. When the root grows old, you can see that the suberin increases around the cells, okay, leaving some cells that are not heavily thickened with suberin, and these cells are called passage cells. So you have heavily thickened with suberin cells, okay. Now, let's come to this drawing here. This is like three-dimensional view appearance of one endodermal cell. This is the lignin. This is the suberin. This is the suberin, all right? Okay, around the wall. Okay, well, what happens is we are going to look at it here first. So this layer is the endodermis. Here, this is the endodermis. This is the endodermis. This is the dicot root. All right, I should have asked which type of root. There's dicot root, the xylem in the middle, the phloem here, and this is the endodermis. You see that these walls are completely enveloped in suberin. But look at this. This is a passage cell. The cell is not completely enveloped in suberin. So we have also here, there are passage cells here. There's a passage cell here. These cells are enveloped. Passage cell here, passage cell, passage cell, passage cell. These are completely enveloped. So in old roots, the suberin is formed thick. So if this was an old root, this is a young root, the suberin will be very, very, very thick. Very, very, very thick. And there will be a few of the cells that does not have the whole cell heavily thickened with suberin, and those are the passage cells. Now, what is the function of the endodermis? Well, we look at this one, the cells are not very well seen. The structure is small, but you see this is the endodermis. You see this is the endodermis here. Now, all the endodermal cells, these suberin, they form a strip And that strip is called Casparian strip. Casparian strip. It's one continuous strip of suberin. All right, good. So the Casparian strip is a waxy substance made up of suberin. So the function of the endodermis is that when water is moving into the xylem from other cells, the Casparian strip will prevent the water from wholesale moving into the xylem. Okay? Now, we'll use this to demonstrate this. Now, this is the suberin around the endodermal cell. Water and gases and other substances that are found in water, when the water is moving across the cell into the xylem, the water and other substances cannot move through the cell wall. And water and substances moving through the cell wall is known as apoplast pathway. Apoplast is APO plast, all right? Apoplast pathway. So water can move through the cell walls. Water can also move from cell to cell through the cytoplasm, all right? And that is simplest pathway. And water can also move from vacuole to vacuole, all right? So vacuole to vacuole is vacuolar pathway. Good. This we will look at it again in a good time. So the endodermis makes sure that as water moves, when it gets to the endodermis, the water cannot move through the cell walls because the cell wall has suberin. It's a waxy substance. Water cannot pass through it. So the substances will have to be forced through the cytoplasm or the vacuole, all right? So that it will control what is moving into the xylem. Let's use this illustration. Yes, let's use this to illustrate the point. This is a stem I have drawn and roots. And this is the root hair. Okay, when water is moved into the root hair by osmosis, the water moves and gets through the epidermis because the root hair is part of the epidermis. So the water moves into the epidermis. The water moves into the cortex, collenchyma and parenchyma. But then this is the endodermis. The red one here is the endodermis. So when the water moves and gets into the endodermis, the water cannot move through the cell walls because of the Casparian strip, because of the suberin, all right? So when it gets into the endodermis, the water will have to move through the cytoplasm or the vacuoles through the plasmodesmata. So the endodermis restricts the flow of water and all other substances through the cell wall. You know the cell wall is really permeable, but because of the Casparian strip, the water will have to pass through the cytoplasm and vacuoles before it gets into the xylem. So the endodermis protects the plant from bad substances like pathogens or mineral salts that are not needed from getting into the xylem because the substances cannot freely move through the cell wall. You know, the cell wall is partially permeable, freely permeable, freely. It allows everything to pass through. But because of the Casparian strip, only substances that the cell needs will be able to pass through the cytoplasm and the vacuole, all right, into the xylem. And then the endodermis also makes sure that substances do not leak from the xylem into the endodermis, into the cortex. So substances in the xylem are also good substances that go in, are protected from leaking back. So the endodermis is very, very, very important structure, and is basically parenchyma that is modified. Good. Now, the other function of parenchyma tissue is because it has a lot of intercellular spaces, it allows gaseous exchange. So you see, anywhere that there are parenchyma tissues, because of the intercellular spaces, the cells are not dead, they are living, and it can keep the plant alive, even where it is in small areas of the plant. So we have, this is a gas space, gas intercellular spaces. You have big intercellular spaces in the leaf, all right? Big intercellular spaces, because the leaf takes in carbon dioxide and oxygen and comes in for photosynthesis. So the leaf needs a lot of intercellular spaces, and the parenchyma in the intercellular spaces have been arranged that way, all right? There are some spongy tissues, like in water plants and in other plants, that have spongy tissue, all right? So those ones, this one is called Aerenchyma. See that it has particularly, all these are holes, and these are found in water plants, okay? You also have these Aerenchyma tissues. You also have these Aerenchyma tissues in other places. We'll look at them in another time. Now, the parenchyma tissue as an epidermis, this is the epidermal cells. These epidermal cells can be modified into a lot of structures: hairs, glands, spines. They all come from the epidermis. You see a plant and you see hairs, it's from the epidermis. We have to look at the epidermis and Aerenchyma tissues again in detail. We have to look at parenchyma. Well, so that's the epidermis. The epidermis has cutin laid on top of it to keep it from losing too much water, desiccation. Now you see this upper epidermis, the upper epidermis, if you see them stained, you will see that they are transparent. The lower epidermis here, they are all transparent except the guard cell. This guard cell and this guard cell, they have chloroplast. And look at the hole that is left, all right, stoma for gas to go through. Well, we'll have a time to look at it again. We'll look at parenchyma tissues as tissues that store pigment and waste, all right? So you have a lot of waste like tannins, glue, latex, all right? They are all stored up in some parenchyma cells, all right? So you have wood. This wood, they all turn brown or black, especially the heartwood. Heartwood here is dark in color because it has a lot of tannins, all right? Tannins. So parenchyma cells store a lot of waste. We'll have to look at parenchyma cells again. It stores water and it stores food, all right? You see these parenchyma cells, these are all parenchyma cells. The part of it inside it, they are starch grains, and starch grains stored in the parenchyma cells of the roots is very common. And the starch grains, also known as statoliths, they can fall by gravity and cause the roots to change, respond to gravity. All right, we have to look at it in detail. Well, this diagram is well lated than the first one. So this is the endodermis. The first one was too small, so we couldn't see the passage cells well. You see, all this is the endodermis. Very lignified walls here. Very lignified. This is a passage cell. You see, this and two. These two are passages. This two passage cell. These two passage. Right. All right. Good. So we want to look at the pericycle now. The pericycle is also a layer that is found in the stem or the roots. In the roots is found closely beneath the endodermis, right? And it turns meristematic. When it turns meristematic, lateral roots can come out of them, okay? So we'll look at it. The pericycle can be one cell thick or many cells also thick. Here's endodermis, here's pericycle. And we saw pericycle as Sclerenchyma in the dicot stems. So we want also to look at cork cells. You see, all here is cork. Here is cork. And cork cells are also parenchyma cells that have taken a lot of suberin inside them. Okay, good. We will have a time to look at these are parenchyma cells that are known as Sclerenchyma because they have chlorophyll to photosynthesize. And you see all these, they are green. These are the parenchyma cells. This is an S-phase. You see that they are green to photosynthesize. This one is not stained to show the green chloroplast. Otherwise, a lot of palisade cells will show the green chloroplast. So parenchyma cells that have chlorophyll that photosynthesize are known as Sclerenchyma. So that is what brings us to the end of today. Thank you and goodbye.